I'm Jeff Hoy. I'm a sugarcane pathologist with the LSU Ag Center. In this presentation, I'm going to provide information about some of the diseases that affect sugarcane and how we control them. And then one of the examples, the last example we'll consider, will be one of the diseases called red rot. It causes rot of the seed cane that we plant, and that's going to be relevant for another big project that we've had that's of interest to the industry, where we're trying to come up with a way to figure out a way to plant billets or stalk sections rather than whole stalks. So any crop is affected by an array of different kinds of diseases, and in the case of sugarcane, we have diseases caused by different kinds of pathogens. You could think about viruses. Viruses cause mosaic, which is a historically very important disease in sugarcane. Our most recent disease is a virus disease called yellow leaf. Bacteria cause ratoon stunting disease, RSD. It's an old foe of the industry. Caused problems for many years. We finally got it on the run. Another is leaf scald. There's some very important diseases caused by fungi. So those include smut, rust, and of course red rot. So these kind of pathogens are in plants just as they're the same thing in people. So with people, if you think about what kind of viruses are causing diseases, you have colds, flu, and of course COVID-19. Bacteria, things like pneumonia, various kinds of infections. We're fortunate, however, that not too many diseases are caused by fungi in people, and that's a good thing because they're pretty tough customers. So when a plant comes under attack from a pathogen, what happens? Well, it could be resistant. So this phenomenon of resistance could limit infection or prevent infection altogether. Alternatively, it's going to limit it in some way, reduce the severity of the infection and the amount of damage it causes to the plant growth and yield. So when you consider a whole population of a crop like sugarcane, you'll probably see a range of levels and types of resistance. And that's true with most of the diseases in sugarcane. And when that's the case, the breeders can go in and they can consider disease resistance to be one of the more important traits. So we think about the crosses that we're making, the parents that we're choosing, whether we're going to have resistance, and we're going to have, during the selection process, work with the pathologists and work together to develop varieties that are resistant to not one but multiple diseases. So that's by far the most favorable outcome for a grower. It has no direct cost or effort on their part. It is something that can be quite effective. So some examples in sugarcane of that would be mosaic and smut. We had a scare a little while back. In 2016, mosaic showed up in some of the varieties that were near release, so we had to do a really intense research project to assess the threat of its reemergence, and you've heard about that over the field days some of the most previous years, and we've figured out ways to ensure that we can screen for resistance and identify susceptibility in the parents that we're using for crossing and continue to develop resistant varieties. But what happens if you have a disease where you don't have good levels of resistance? There are no resistance mechanisms that are effective. Well, if that's the case, you got to come up with some different approaches. And that's going to depend on, well, the type of disease and factors at the field level, like how does it spread and increase. So examples here, back to RSD. It's caused by bacteria in the water vessels in the stalk that build up during the season, and they're spread by the harvester mechanically. Well, there are no good levels of resistance to that out there, and so we've had to manage it in a different way. And what we ended up doing there is eventually we were able to control it by a healthy seed cane program. So now we're using a lab process called tissue culture to produce healthy planting material for the growers, and it's been pretty effective. It's something altogether, it's a public and private sector partnership, and I've talked about that in the past, so it's called seed cane certification. Another example is, I think, a fungal disease, brown rust. It's caused by a fungus that infects the leaves, causes those red-brown lesions, and when it gets on the young leaves and it gets bad, the whole field will look red. Well, we tried to control that with resistance, but it doesn't last. The pathogen is very adaptable. So when you put a variety out there that's resistant in the field and the acreage starts going up, the fungus adapts and overcomes that resistance. So we actually have a major ongoing research project to try to still come up with resistance that will be effective. It's something we call molecular markers for resistance that we're looking for. So you're looking for DNA segments that can have linkage to resistance genes in the host, and that's going to in turn allow you to track them with a lab test. Another example is brown rust. It's caused by a fungus that infects the leaves and causes a lot of lesions with this red-brown color. And when it's bad, it gets on the young leaves and builds up. The whole field looks red. So we've tried to control this disease with resistance and found some, but it's not durable. We've put it out in the field with a resistant variety, and the pathogen is so adaptable that as the acreage starts to go up, it adapts and it overcomes the resistance. We are trying to come up with a way to make resistance more effective. There's an ongoing major research project. We're trying to develop what are called molecular markers for resistance. And this is going to be DNA segments that are closely linked with resistance genes. And if we can find some of those that are effective enough, we can actually track those in the plants and the parents and make crosses to make combinations of genes that we think will be more effective and durable. So we have some hope for that. But in the meantime, we've gone with a more direct approach. We're just using fungicides to control the disease, and we found some that are effective. So when the disease occurs in the spring, they can minimize the damage it causes, and we've been able to grow some high-yielding varieties with susceptibility, just manage the disease when it does occur. And that's an ongoing process. We have some new products in the last year or so. We've come up with a couple of Triva Pro and Revitec. So we're in pretty good shape there until we can come up with a better solution with resistance. The last example would be red rot. So it's a stalk rot, and in our case it affects the seed cane after it's planted. So our seed cane goes into the ground, and then it's affected potentially by adverse environmental conditions, and they interact with these stalk rots, of which red rot's the worst. So you end up with some real stand establishment problems and survival. Unfortunately, the billets, these stalk sections that we would like to plant, are more sensitive to the problem and more severely affected. So in this case, we've had to take some very different approaches. We're just trying to avoid the conditions that lead to problems. So cultural practices have turned out to be important. So we're planting longer billets with 3 or 4 buds per billet, and that's helping along with less mechanical damage caused by physical damage from the harvester, and now we're looking at the planter. So these are less wounds for where the disease can get started. So couple that with good planting practices. Good soil moisture is very critical. Dry conditions after planting, you can't control that, but they can lead to a real acceleration in the severity of red rot. So if you have good soil moisture to start, you avoid those conditions. We are looking at more direct control measures. We're looking at fungicides. It's the same fungicides that we're controlling rust with, and seeing some benefits there. So that's going to be a help. We're interested in another chemical. It's an insecticide called platinum or thiamethoxam, and this is a very interesting chemical. It goes very systemic in the plant. When you treat the billet, the plant starts to grow, it takes up the insecticide, it moves throughout the plant, and it causes a change in the physiology of the plant. Some other plants they've figured out is something they call a phenomenon called induced resistance. So this is one of those cases where it's not total resistance, but it's limiting the damage that's occurring. And in this case, though, it's an even broader effect. It can have effect on maybe we call it biotic stress diseases, but also abiotic stresses like the drought stress. So that sounds like exactly our combination that's a real problem with billets. So we are seeing some real benefits from this treatment, particularly in combination with the fungicide. Unfortunately, we haven't gotten a label for that insecticide. It's what they call a neonicotinoid, and they're very successful because they're so effective, but a lot goes out, and it's been implicated in what they call this bee colony collapse. So we're having trouble getting a label, so I'm not sure what's going to happen there. We're continuing to work on it. Have a student project that's trying to get a better handle on what's actually happening in those billets down there in the ground, and she's actually looking at a gene expression study to see what kind of resistance mechanisms are going on. And we'll keep working on that. And but in the meantime, this other combination of things that we've pulled together, different approaches all together, is helping a lot. So growers are having success with billet planting. There are even when adverse conditions occur that could lead to trouble in the past. So we're optimistic that we may be able to, more growers are switching over, and we may be seeing a transition over that'll allow us to switch to billet planting. So that's about all we have time for today. I hope it's been worthwhile for you, and I thank you for your attention.